today, since there was no repro on there, it's not my fault. So you guys can wait for that for the final exam. Okay, so let's do a quick recap of what we talked about yesterday. I know with this exam in between, it was maybe some of this stuff got diluted out a little bit. So what did we talk about yesterday at noon? Wow, that's kind of a loaded question. What the heck is happening?
We talked a lot about like sex determination as well as gonads.
Yep, perfect. I love it. Sex determination. We talked about sex determination. So really, we decided that sex determination means all the processes that are involved in an XY individual developing testes and looking like a male, and an XX individual developing ovaries and looking like a female. Okay, so sex determination. What are the three processes that underlie the process of sex determination? That's the first one. Go for it. Chromosomal sex determination. Really, all that means is that the sex chromosomes are going to decide. Okay, this is the point when we have sex chromosomes getting together, and we're either going to be XX or we're going to be XY. That's it. Okay. When does that happen? I should have brought a gigantic laser pointer and just start pointing at people. At conception, at fertilization, right? As soon as we fertilize that egg has 22 autosomes, numbered 1 through 22, that's it. And then an X chromosome, that's it. Okay? And then the sperm will either bring 22 autosome and an X, or 22 autosome and a Y chromosome. That's it. So at that point of fertilization, now we have an embryo that starts as XX or XY. And what's the second part? After that we have? Gonadal. Yeah, gonadal sex determination, okay? And another word for that was, what did I want you to write next to it? Primary, okay? So gonadal or primary sex determination. So what the heck does that mean?
Whether you have ovaries or testes?
Yeah, so basically we have this structure or organ with no identity, so we're going to call it a gonad. And it's bipotential, a bipotential gonad. And during gonadal sex determination or primary sex determination, we're actually going to give it an identity. So if at chromosomal sex determination it was determined you are XX, then that gonad will turn into an ovary. Okay? And if during chromosomal sex determination you were destined to be XY, then your gonad is going to be a testis. Okay? So the last part of sex determination then is what? Phenotypic or secondary sex determination. Okay? And that process, again, happens only after primary or gonadal sex determination. And that involved what? There were two parts to this. What was phenotypic sex determination?
Starting to develop the internal reproductive tract.
Perfect. So we start, the first part is developing of male internal reproductive structures or female internal reproductive structures, right? And then the second part would be external genitalia, right? So actually externally looking as a male or a female. Okay, so that together is secondary or phenotypic sex determination. Okay? Make sense? Any questions about that stuff? Perfect. Okay. So this is where we finished then, right? So I told you yesterday that I really want you to know those 5 factors that are listed on this slide. Okay. And then there are 4 that are labeled in red. They are involved with the ovarian developmental pathway, and there are 2 labeled in blue that are the testis developmental pathway. Okay. So these factors are absolutely necessary in order for this to happen. So they're all part of what? Which of the 3? Chromosomal, gonadal, or phenotypic? So what is shown right here is what? Gonadal, okay? Are any of these hormones? No. Remember I told you, gonadal sex determination, after that, next to it, I wanted you to write genetic factors. Phenotypic sex determination, I told you to write next to it hormones, okay? But we're not doing that here right now, okay? Of those 5 factors that I want you to be able to at least recognize on the exam, how many or which one is present on sex chromosomes?
SRY.
SRY, and it's present on the Y, okay? Any other ones? No, okay, that's the only one. So we talked about the fact that this is kind of a potential problem, right? The fact that you have 4 of these things, 3 on the female side and 1 on the male side, that is present on an autosome, what is a potential problem? Let's look at the difference. Does everybody in this room have a Y chromosome? No. Does everybody in this room have chromosome 1 through 22? Yes. Okay? Okay, so now think about this. Those other 4 factors that are not on a sex chromosome, that means everybody has that. Whether you're male or female, you have that. Okay? That is a problem. During development, do I want RSPO1 Win4 beta-catenin to be turned on? I'm talking about myself. No, that doesn't work for me. I'm XY, okay? It would interfere with testis development. So what prevented these 3 factors, which I do have, they're on my autosomes, what prevented those 3 factors from being turned on in that particular stage of my life?
Well, when you have SRY or the SOX9, it inhibits the production of those genome matter.
Perfect, exactly. I had SOX9 turned on because of SRY that prevented these factors from being turned on. Okay, does that make sense? So vice versa, we're going to do it the other way around too. If you're XX and you're biologically destined to develop ovaries, a potential problem would be if somehow SOX9 was turned on because you have that as well. So what prevented SOX9 then from being turned on? RSPO1, Win4, and beta-catenin. Okay. So we call these antagonizing developmental pathways. So the testis developmental pathway and the ovarian developmental pathway are antagonistic. They antagonize each other, they inhibit each other. Okay? Make sense? I hope so. Because that was kind of like a 8-minute recap of yesterday's 50-minute lecture. Wow, it can get really efficient once we have slides and really get focused. Okay. Before we get going on, because today we're going to wrap up gonadal sex determination, then we do phenotypic sex determination. Are there any questions about this stuff? Make sense? Good. Okay, this slide I made late yesterday, and so it's not posted, but we'll put this on the board, okay? So don't worry. Basically what I want to do right now, I want to focus again on the cells that are inside the bipotential gonad, okay? I told you yesterday, reproductive physiology is not inherent conceptually very difficult. Like it's not about action potentials and things, pluses and minuses and something that you can, okay? It's nomenclature, okay? We give cells names and we give tissues names and we have hormones going some places, that's it, okay? So here is a bipotential, undifferentiated, identity-less gonad, okay? And here we have the mesonephros. Yesterday I made the analogy of a hot dog on a bun. The gonad is the hot dog, the bun is the mesonephros. 2 separate structures, but they do work together, okay? So no identity here. When it has no identity, one of the cell types that are present are called somatic support cells. Okay, somatic support cell. What is a somatic cell? I guess I beat everybody to that question. about it this way. An average adult body has how many cells? This is just stupid trivia, but you may look really smart in front of your family and relatives. 37.2 trillion, okay? Those 37.2 trillion cells are all packaged into tissues and organs and everything packed together into a body. So basically the body is made up of two primary cell types, two. One is somatic and the other one is what? It's the only cell type that can do meiosis. That's a hint. So what is that second cell type?
Sex cells?
Yeah, the germ cells, sex cells. Okay? So what is a somatic cell? Well, all the cells that are not sex cells or germ cells, those are somatic cells. Okay? So the first population of cells that we need to talk about that exist in that bipotential or gonad without identity is the somatic support cell. At some point, if you're XX or XY, this cell is going to differentiate. It's going to establish an identity. Okay? And this somatic support cell will turn into Sertoli cells in the testis, or XY, and granulosa cells in the ovary. Okay? And the reason why we're doing this is because I want you to recognize now that at some point we're going to talk about an adult ovary and an adult testis, and you have all these different cell types. We're going to be talking about Sertoli cells. We're going to be talking about granulosa cells. Basically, what this is telling you that early on, these cells actually come from the same origin. These were the somatic support cells. Okay? Now, the factors, the genetic factors that we just talked about, these 5, are extremely important for these to develop first. Okay? So once we have SRY SOX9 turned on and RSP1-Wnt4 beta-catenin turned on, this is happening. Okay? So this is due to SRY SOX9, and here are the other 3 factors. Okay? These cells differentiate first. First one to do that. Now, there's another somatic cell population in the gonad. They're called precursor steroidogenic cells. Okay. What could this cell population be doing later on when they're mature? If you look at the name.
Produce hormones?
Yeah, but specifically. Steroids. So these are steroid-producing cells. Okay? We call them precursor now because early on they're not doing anything. They are destined to become steroid-producing cells. Okay? In the testis, these are called Leydig cells. Okay? And in the ovary, these are theca cells. And in fact, the major steroid hormone that it produces would be testosterone in both cases. Okay? So yes, females also make testosterone. Females also have testosterone floating around in the bloodstream. Testosterone is also necessary in order for female reproduction to happen. Okay? So we'll talk about that later. This, however, has to happen first. Once this happens, the first somatic support cell differentiate first, then this can happen. Okay? And then we finally have one more population of cells that are called primordial germ cells. Okay? Primordial germ cells. Okay, so these are actually the germ cells. So we have somatic cells and germ cells. So finally, here are the germ cells. These, at some point in the adult, will develop into spermatogonia, okay, in male, in testis, and oogonia in the ovary, okay? So that's it. That's kind of the anatomy of a bipotential gonad. When it starts developing in testis, we have these cells, and the ovary, we have those cells, okay? Make sense? Awesome. Okay. The only thing we have to recognize is when we talk about a bipotential gonad, so at this stage, okay, these are the only cell types that are present. Germ cells are not there. Okay? And in fact, what has to happen is germ cells actually develop somewhere else. They have to migrate towards this bipotential gonad. Okay? So when all this stuff is going on with these genetic factors in these somatic cells, right, that's around the same time that germ cells start populating this tissue. Okay? In mice, it takes about 48 hours. In humans, it takes a little bit longer for this whole process to happen. Okay? So this is just a picture showing a 3-week and a 4-week-old human embryo. And so the heart is right here. So this would be the head region. This is the other end. And this would be a gigantic yolk sac. And if you look at this picture, there are just special signals that are secreted by surrounding cells that cause a few cells, somatic cells, to switch identity and turn into germ cells. It's pretty amazing. Okay, the body actually does this normally. So these germ cells actually develop outside of the embryo, near the yolk sac. Okay, and then between the 3rd and the 4th week of gestation, these germ cells that have formed now, they need to start migrating. Okay, so develop around here, and they migrate along the hindgut all the way up towards the gonadal ridges or to the bipotential gonads. Okay, and then when this stuff starts happening, they start populating that gonad, and eventually this happens. Okay, make sense? So we are still during fetal life. Okay, we're still fetuses. Okay. Okay, so what then is involved with this process? How does this germ cell know to develop as a male germ cell or a female germ cell? This has nothing to do with sex chromosomes, absolutely nothing. Okay? What this has to do with the surroundings. The germ cells start populating the gonad, and whatever the gonad is going to turn into will tell the germ cells what to become. Okay? So, let's take one step back. When we do all this stuff, we end up on this side with a gonad. So I'm taking, okay, the mesonephros right here. Let's focus on the gonad. This is a testis, and it has these cord-like structures inside. Okay? That is a fetal testis during fetal life. Now it has an identity. You have Sertoli cells, Leydig cells, all that stuff. In this end, when this process is done, we kind of have this odd structure that has no, well, tissue or organ, an ovary, but it has no structure to it. Think of it just as a bag of marbles with different colors in it. Each color is a cell type. But it's not organized. It's just randomly distributed. Okay? One of these cords, let's highlight one. Okay? What we find inside, all the way inside, are germ cells. Okay, so this is GC for germ cell. Okay? And surrounding these germ cells are Sertoli cells, SC, Sertoli cells. So basically each of these cords have Sertoli cells that wrap around germ cells. Okay? In between these cords we'll have Leydig cells, but you could ignore that for now. Okay? Why is this important? It kind of pertains to this slide right here. The mesonephros, whether you're a testis, actually, let me do this one right here. So mesonephros, gonad, whether you're XX or XY, doesn't matter. In both cases, the mesonephros secretes retinoic acid or vitamin A. Okay? The mesonephros just does that. I don't know, it's like continue with a stupid analogy now, is maybe the bun is laced with melted cheese and it percolates into the hot dog. Okay? It kind of sounds disgusting, but if germ cells, doesn't matter if this is an XX germ cell or an XY germ cell, in general, when germ cells see retinoic acid, they're exposed to retinoic acid, they enter meiosis 1. You have to write this down. You have to know this. We're going to talk about meiosis in the ovary. Anything that we're going to talk about next week with the ovary will not make any sense if you don't have a grasp of this part. Okay? You need to know that in the fetal ovary, because of its exposure to retinoic acid, the germ cells enter meiosis 1 and arrest in meiosis 1. So that is female differentiation of germ cells. That means in the ovary, during fetal life, all the germ cells in the ovary enter meiosis 1 and arrest in meiosis 1. So when a baby girl is born, she normally has 2 ovaries, and all her eggs are arrested in meiosis 1, because it happened early on in fetal life. Okay? Because of the exposure to retinoic acid. Now, what happens in the testis? Well, something completely different. Okay? What's important to recognize here is that these Sertoli cells that wrap around these germ cells have this enzyme with an absolutely atrocious name called CYP26B1. Okay? This enzyme gets rid of retinoic acid. It gets rid of it. It destroys retinoic acid. Okay? So, what does that mean for these germ cells that are surrounded by these Sertoli cells? Do germ cells in the fetal testis see retinoic acid? Yes or no? No. The Sertoli cells get rid of the retinoic acid because of that enzyme. They have that enzyme. Okay? Well, what does that mean? Well, that means that the germ cells in the testis do not enter meiosis 1 during fetal life. Instead, they continue to proliferate, or mitosis, which is proliferating, for a little while, and then they just kind of stop. That's it. So if there's a question on the exam that talks about differentiation of germ cells, female or male, the key difference here is what?
Mitosis 1.
Well, yeah, but what is in general the key difference between a female germ cell and a male germ cell during fetal life? What happens to them?
Exposure of retinoic acid.
Yeah, and that causes?
The presence or not of CYP26B1.
Yeah, but but the exposure to retinoic acid, what does that do to the germ cell?
Gives the female ovary the eggs.
Yeah, but specifically?
It allows them to go through meiosis.
Meiosis, 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 meiosis. It causes them to enter meiosis. And that's a big thing. That has something to do with the becoming haploid. That's what germ cells do. That's the whole point of sexual reproduction. Okay? So they enter meiosis. Perfect. Now, in the male germ cell differentiation, do they enter meiosis? Yes or no?
No.
Not during fetal life. In the adult, yes. In fetal life, no. Why do male germ cells not enter meiosis in fetal life? Because of this stupid enzyme called CYP26B1. Make sense? Okay, let's do it this way. Let's say there's a question on the exam. There's a male individual or a fetus, and it has a mutation in CYP26B1, a mutation such that it doesn't work. What happens to the germ cells in that testis? They would enter meiosis 1. Yeah, so just like what would happen in a female, right? Now, let's say this is really a crazy-ass mutation, and now we're an XX baby with an ovary, but a crazy mutation that causes CYP26B1 to be turned on in all the cells. What happens to the germ cells? Doesn't enter meiosis, because the CYP26B1 gets rid of retinoic acid, it cannot enter meiosis. Make sense? Perfect. Okay, so you can ignore the bottom part here, gonad sex differentiation. Basically that says whether a gonad develops as a testis or an ovary has nothing to do about what the germ cells are doing. It's the other way around, right? First we make Sertoli cells because of these genetic factors, making a testis and these cords, and that will determine whether the cell goes into meiosis or not. You can actually take XX germ cells, and if you would shove them into a testis, what do you think is going to happen to that XX germ cell? It's not going to enter meiosis because of CYP26B1. Make sense? So that's kind of that bottom statement. Okay, so then we finally, to kind of summarize what happens, on the top it says that basically in the fetal, making a fetal testis or testis development involves development of vasculature. That's kind of the key thing. Ignore the proliferation and blah, 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 it doesn't matter. Vasculature development is kind of important because remember when we talked about hormones? What does hormone and vasculature have to do with each other? Well, we can only have hormones if there's vasculature. Hormones is a chemical substance released in the bloodstream, goes places, okay? The other thing is that germ cells in the testis, they do not enter meiosis. They continue to do mitosis, which is just cell proliferation. They divide. And then at some point they enter mitotic arrest, which just basically means they stop proliferating after a while. Really, by the time a baby is born, male or female, up until puberty, there's nothing interesting going on. Okay, that's the least interesting part of life related to reproductive physiology. In females, however, germ cells enter meiosis, meiosis 1, and you have to write down or at least know that they enter meiosis 1 and arrest in meiosis 1. They stop in meiosis 1. Okay. How many of you have heard of this concept or dogma that a baby girl is born with all the eggs she will ever have? Most of you? Has to do with this, right? These germ cells enter meiosis 1, arrest in meiosis 1, and that's it. Okay? Any questions about this stuff? Cool. So what we're going to do now is we're going to focus. So basically we wrapped up primary orgonatal sex determination. We're going to do the last part. So we're going to focus on the mesonephros now. So the mesonephros contains 2 ducts. So the gonad is bipotential. Apparently the mesonephros is bipotential too. Because whether you're a boy or a girl, early on in fetal life, you had a male duct and you had a female duct. Okay, so something needs to happen. That is secondary or phenotypic sex differentiation. So the female duct is called the Müllerian duct. It's in the mesonephros. The male duct is called the Wolffian duct. Okay? Think of this as just a long tube. And this long tube, whether it's male or female, is going to give, we're going to start naming this long tube in different parts. And all these different parts get a different name. Okay? So, in the case of the Wolffian duct, the part of the tube that's closest to the testis is going to give rise to what is called the epididymis. Okay? So this is just all nomenclature. This is just names. Okay? So the Wolffian duct develops, the first part is the epididymis. The second part is called the vas deferens. And the last part of the duct gives rise to an accessory sex gland, which is called seminal vesicle. Okay? So you guys are well experienced in multiple choice exams and things like that at this point. Can you imagine how easy it is to come up with a question like this? Right? Which of the following structure comes from the Wolffian duct? A, vas deferens, B, kidney, C, neurons, or D, adrenal gland? Everybody's thinking, wow, if that dude comes up with these kinds of questions, I'm going to be golden. Just think about this a little bit, okay? So just know the different parts that the Wolffian duct gives rise to, okay? Now, let's do the female. The Müllerian duct gives rise to the oviduct, okay? The next part of that tube is the uterus. The next part of that tube is the cervix. And the last part of the tube is the most inner portion or upper portion of the vagina. Not the whole vagina, but the uppermost inner portion. Okay? So again, those you need to know those structures and recognize they all come from the Müllerian duct. Does the ovary come from the Müllerian duct? Yes or no? No. It's not like the bun comes from a hot dog. These are separate things. Okay? Now, how many of you have taken an anatomy class? A couple. Excellent. So if you look at these figures, I want you to focus on the female part. Anything odd? Go for it. Sorry? It looks like a tube. Like which part specifically looks odd of the female reproductive tract? And see guys thinking, now what, there's something off? Isn't that what it's not supposed to look like? The uterus. The uterus is kind of odd here, right? Because it's shown as uterine horns. Yes, rodents and sheep and cows, okay, they will have uterine horns. Women here do not have uterine horns. It's a simple simplex uterus, okay? This figure just comes from a review paper that mostly focused on rodents. So I'll show a proper picture of the female reproductive tract next week, okay? Okay, so what's going on here? Here we're going to meet our friend Alfred Jost, a French endocrinologist with very steady hands. He did a lot of crazy ass stuff. Okay, basically what he did was fetal surgeries. Okay, so we're going to talk about his experiments a little bit because his experiments led the foundation to our understanding of how these ducts develop and what is involved with the, for example, at 20 week of gestation, how a boy looks like a boy and a girl looks like a girl, the external genitalia. Okay, so the kind of experiments he did mostly on rabbits, and he did fetal surgeries. Okay, so this picture that you're seeing here is actually one of his fetal surgeries that he did. He had pregnant rabbits, and he would make a small incision in one of the uterine horns, expose the fetus, and then remove the gonad. And then he would suture everything back up and just let the pregnancy proceed and the baby bunnies be born, and then just kind of ask questions. Okay. In fetal life, I removed the gonad. What impact did this have on the reproductive structures? Okay. So let's go through his experiments a little bit. Okay. So the legend for this is on your left. The Wolffian duct is blue. The Mullerian duct is purple or pink. Recognize again, I want you to know what these structures give rise to. If we talk about the Mullerian duct, I want you to know oviduct, uterus, cervix, upper portion of vagina. Okay. So the first experiment that he did, and we'll go through these experiments and then see what happens in XX and XY. Okay? In the case of XX, he removed an ovary. In the case of XY, he removed the fetal testis. So in the first experiment, what happened was this. Okay? So he removed the fetal gonad. In the case of XX, what he found was the Mullerian duct was there. So basically, after birth, these bunnies had a uterus, an oviduct, a cervix, and an upper portion of vagina. Okay? The XY bunnies had the same thing. Is that normal? No. Okay? So that was very confusing. The next experiment, what he did the same thing again, removed the gonads, but at this point, he had these little capsules that were laced with testosterone, and he put that quickly back in. So basically, he was replacing the gonads with a capsule that released testosterone continuously. Okay, so let's see what happened here. This is what happened. Now we had bunnies, whether they were XX or XY, that had the female internal reproductive tract and the male internal reproductive tract. So what is the difference in the experimental design of experiment 1 and experiment 2? Yeah, the presence of testosterone, right? So in both cases, he got rid of gonads, but in experiment 2, he put testosterone back in. What is the difference in the results that he got?
The expression of both male and female reproductive tract.
Yeah, just very specifically, what is the one difference between experiment 1 and experiment 2 in the outcome?
The addition of the male reproductive?
Yeah, so the Wolffian duct is there. That's the one difference, right? So what conclusion do you think he made?
So could you say that testosterone dictates the formation of the Wolffian duct?
That's it. The one difference in the experimental design was the addition of testosterone. The one change in the outcome was the presence of the Wolffian duct. So he concluded that testosterone was necessary for the Wolffian duct to exist and to develop into the male internal structures. Right? Does that make sense? Yes? No? If not, say something now because we're going to keep going. Good. Okay, last experiment, what he did was he did the same thing again, removed the gonads, and now he added a piece of testis back. Okay, a whole testis or a fragment of a testis. And then we're going to look at what happened with XX. We don't have to figure out what happens with XY because that's an experiment where I remove a testis and I put a testis back. That's just mean. Okay, there's no point to doing that. Okay, so let's see what happens with XX. This is what happened. Okay, so if you look at this one, now we have gonad removed, and we put a piece of testis back, and the only thing that develops is the male internal reproductive tract. What did we need for the male internal reproductive tract? What was the thing that we needed? Testosterone. So this means that testosterone coming from the testis causes development of the Wolffian duct, right? Now, what is the other thing that we're seeing here? We see the Mullerian duct is gone. That also means that the testis secretes something that causes the Mullerian duct to disappear, right? Does that make sense? And in fact, because of that experiment, he identified a hormone coming from the testis that is called anti-Mullerian hormone. Okay, so AMH. AMH is anti-Mullerian hormone. This is a peptide hormone. It has nothing to do with testosterone. The only thing testosterone and AMH have in common is that they come from the testis. That's it. Testosterone is a steroid hormone. AMH is a peptide hormone. Okay? AMH comes from Sertoli cells. Okay? Testosterone comes from the Leydig cells. Okay? Make sense? Makes sense to me, but yes. Anti-Mullerian hormone. The nice thing with that name is that hormone tells you exactly what it does. Okay? So it's kind of summarized on this slide right here, testosterone, AMH. Okay? So on the normal condition, first we have establishment of XY embryo. All the cells become XY. We have a bipotential gonad that is XY. SRY and SOX9 get turned on. We're making Sertoli cells. Then we make Leydig cells. Sertoli cells secrete AMH, gets rid of the Mullerian duct. Testosterone secreted by Leydig cells maintains the Wolffian duct. That's it. What about the ovary part? Well, the key difference between the fetal testis and the fetal ovary is, aside from enteromeiosis for germ cells and mitotic arrest in testis, is the fact that the testis secretes hormones and the fetal ovary does not. The fetal ovary does not secrete any hormones. Okay? What would happen if a fetal ovary secretes testosterone? What would happen in terms of the internal plumbing? Yeah, you would have the Wolffian duct and the Mullerian duct. Make sense? Good. Perfect. Okay, that's the first part of secondary or phenotypic sex differentiation. This is the second part, okay, external genitalia. You already knew before taking this class that you had to wait a period of time in order to assign sex to a baby by ultrasound, because before that they all look the same, okay? So again, the external genitalia are bipotential. They have the ability to go one of two ways, okay? Initially, the bipotential external genitalia have a genital tubercle, a labioscrotal swelling, and a urethral groove, okay? What I want you to write next to this box is the presence of that they contain androgen receptors. So the external genitalia, when they're bipotential, have androgen receptors. Whether you're XX or XY, it does not matter. Androgen receptors are the receptors for testosterone, or any testosterone like steroid. Okay, we call them androgens. Okay. The other thing that this external genitalia has when it's bipotential is something called 5-alpha reductase. Ends with an ACE, so must be an enzyme. Okay, so this is an enzyme. Okay, so just write that next to it. Okay, what happens with the male pathway? In the male pathway, the genital tubercle elongates and actually develops into the penis. Okay, the labial scrotal swelling enlarges and develops the scrotum. And the urethral groove actually fuses and closes completely. If it doesn't close all the way, then you have something called hypospadias. Okay. And along the female pathway, the genital tubercle does not elongate, but kind of invaginates a little bit and actually gives rise to the clitoris. Okay, so the clitoris and penis are each other's developmental counterparts. Okay. There's no swelling or enlargement of the labial scrotal swelling. Okay. So that's kind of the male and the female developmental pathways. So how do we get development of the male external genitalia? Well, it starts again with hormones. Testosterone from the fetal Leydig cells are secreted, travels around through the bloodstream, goes towards the bipotential gonad. The bipotential gonad has 5-alpha reductase. This enzyme converts testosterone in dihydrotestosterone. That's the stuff on the bottom there. Okay. Think of dihydrotestosterone or DHT as super testosterone. It works just like testosterone, but way better. Okay. So you need fewer molecules, and when it binds the same receptor, it already elicits a response. Okay. On top of that, we have androgen receptors, so dihydrotestosterone binds the androgen receptors and causes masculinization of the external genitalia. Okay. In the female, do we have any testosterone secretion? Do fetal ovaries secrete testosterone? No. Okay. So we do not have elongation of the genital tubercle. We don't have masculinization of the external genitalia. Okay. Now, let's look at a disorder of sex development. Okay. There's this one. In this case, we have somebody that is 46XY and has complete androgen insensitivity syndrome. I don't want to talk about that one right now. I want to talk about this one. This is an XX infant, okay, with masculinized external genitalia. If I would have taken away the box on top, okay, so pretend you didn't read that, and you just look at that picture, and that is a baby that is born. What sex do you assign to this baby?
Male.
Male? Well, there's really kind of two possibilities, right? Yeah?
Maybe whatever's inside. Whatever they formed inside, if there's like
Ovaries or testes or things like that. Yeah, so, but the thing is, remember, we kind of all know what happens during the delivery, right? Kind of. So that's if we're waiting, boy or girl, we're waiting for screaming. That's actually way more important. Like you want the baby to scream. It's a boy, it's a girl, right? Okay, so there's two possibilities here. You already know what it is. It's XX. But can you imagine that in this case, you're either thinking, okay, this is either an XX that is masculinized, or it's an XY that is under-masculinized. Because trust me, a baby boy doesn't look like that. Okay? So those are your two options. Okay? So what's the first thing that you want to find out from this baby? Think a bit about the order of sex determination. What would be the most helpful to find out first? Even before that, like let's say testosterone, but even before that. Chromosomes. You want to know, is this baby XX or XY? Okay. In this case, the baby is XX. So now we need to figure out how can we explain this. Well, as you see on the bottom, maybe, this baby actually has something called congenital adrenal hyperplasia. Okay, that basically means congenital adrenal hyperplasia means that your adrenal glands are overactive. Do we remember from the endocrine lectures at all the adrenal glands? A little bit. You had a medulla and a cortex. The medulla secreted epinephrine, This is helping you with your comprehensive, awesome exam, right? And the cortex secreted some other crap, right? What is one of the things that was maybe on the exam last night that came from the adrenal cortex? Somebody said it here. Aldosterone. Aldosterone. Yep, that's one thing. What kind of hormone is aldosterone? Steroid hormone. Okay, what's another hormone? Steroid hormone that comes from the adrenal cortex. People think, crap, there's another one? Yeah, there's actually 3. Cortisol, glucocorticoids, and then one more that's interesting here. Starts with a T and finishes with testosterone. Testosterone, yes, very good. Testosterone, androgens, okay? So, can you imagine that if your XX and your adrenal cortex is hyperplastic or is overactive and pumps out lots of testosterone, that this is going to affect external development of the external genitalia. Why? Well, because of this enzyme, 5-alpha reductase that's there. It's going to get converted into dihydrotestosterone. There are androgen receptors there. So it's going to push it towards the male developmental pathway. Does that make sense? Why not completely masculinize? Well, probably because of the testosterone levels were not super high. Okay, at some point you need plenty of testosterone to be converted to dihydrotestosterone in order for complete full masculinization. But in this case, there was enough testosterone to start masculinizing the external genitalia. Does this kind of make sense? Okay, so what I want you to do, I hate giving homework assignments, but this will really help because all these things you can explain if you understand that one slide with a chronological order of sex determination, and if you understand Alfred Yost's experiments. Okay, so this is the same as, or similar, but the same disorder as this one. Okay. So for this one, and I'm pretty sure I posted this, right? For people that have a handout, did I post this one? Good, good, perfect. So this is an individual that has complete androgen insensitivity syndrome. That means the androgen receptor is mutated and it's not working. Okay, it might as well not be there. Okay. Now, I'll already help you out a little bit because we already saw from this slide, what are the sex chromosomes of this individual? XY. Would you, if you would have seen this individual, would you have guessed this would be an XY? No. Okay. So clearly you kind of figured this is probably not an XX. If this is an XX, I'm just showing a picture of a naked woman in class. I may get in trouble for that. Okay, make sense? So, what I want you to do, just to kind of figure out for yourself, write down what do you think this individual has for gonads? Does this individual have any gonads? Does it have testes? Does it have ovaries? Does it have testes and ovaries or an ovotestis? Okay. And then what do you think about the internal plumbing? Okay. Which are present? Is there a uterus? Is there epididymis and vas deferens? Based on what we talked about, you should be able to figure this stuff out. Okay? But we'll do tomorrow at the start of class. We'll go through this quickly and see if we got it all right. If you just practice this at home, at least you'll figure out for yourself if you get this or not. Okay? But we'll go through this in class tomorrow. Okay, good luck today. See you guys tomorrow.